So, good afternoon, students. In previous class, we had discussed about the eukaryotic cell under the chapter cell, the unit of life. In eukaryotic cell, we had discussed about the eukaryotes includes all the protists, plants, animals, and fungi. And there I told you all the eukaryotic cells are various, in the sense not similar. In some eukaryotic cell you are going to find the cell wall, and in some cell wall is absent. Okay, eukaryotic cell consists of plasma membrane or a cell membrane, and inside the eukaryotic cell you are going to find the different kinds of organelles, and those are membrane bound organelles, non-membrane bound organelles, and you all you all know about the what we are going to call it as a membrane bound organelles and non-membrane bound organelles. Okay, so in today's class under the cell, the unit of life, we are going to discuss in detail regarding the cell membrane. Cell membrane, or the structure of cell membrane, it is studied after the discovery of electron microscope in 1950, and that is in red blood cells, human RBC. Okay, so here in cell membrane, it is composed of, or a cell membrane, it is made up of lipids, and these lipids are arranged in a bilayer, in the sense two layers. They are arranged in a two layers. Okay, and these lipids are arranged within the membrane with two, what we are going to call it as the heads. Okay, tail and head. Here I written the lipids are arranged within the membrane with the hydrophilic polar bond, hydrophilic polar end towards the outside, and hydrophobic non-polar head towards the inner side. And here, what is meant by hydrophobic and hydrophilic? Hydrophilic is nothing but water loving. Hydrophobic in a sense water hating, in a sense water repelling, right? These hydrophilic non-polar heads, non-polar tail of a saturated hydrocarbons is protected from the aqueous environment. These, whatever the non-polar tails are there, those are going to protect the cell membrane from the aqueous environment. Aqueous environment protect ಮಾಡ್ತವೆ. The cell membrane also consists of proteins and carbohydrates. And the ratio of these proteins and lipids are varies from one cell to the another cell. In the sense, in human RBCs, you are going to find 52% of proteins and 40% of lipids. 52% of protein and 40% of lipids. Okay, next, the membrane proteins. Again, how we are studied means we are discussed regarding the lipids. They are arranged within the membrane. Likewise, the proteins are also arranged, maybe in the sense arranged in a what we are going to call it as a cell membrane. And based upon their position, there are the two types of membrane proteins. Two types of proteins. What are those in the sense? The membrane proteins may be integral and peripheral. What is meant by peripheral? Peripheral proteins lies on a surface of the membrane, while integral proteins can be partially or totally buried into the membrane. I used to draw the diagram now. Next, immediately, that time I will show you these all the things. Right? This is about the basic structure, which is means regarding the cell membrane after the discovery of electron microscope. And after this, there were two scientists named Singer and Nicholson. These two scientists are improved the model of structure of a plasma membrane, and they are given the name of the improved model of their structure of plasma membrane, that is the fluid mosaic model in 1972. So these are the basic points regarding the cell membrane. Now I used to draw the diagram, and in diagram I used to show you the different labelings regarding the lipid, then bilayer, then I used to show you the polar and non-polar heads, then I used to show you the what we are going to call it as proteins, integral proteins, peripheral proteins, etc. When you are going to draw the diagram, that is very easy to explain what we are going to call it as a cell membrane. Explain the cell membrane. Okay, I will draw the diagram. I used to draw a simple diagram regarding what fluid mosaic model or a cell membrane, bilipid cell membrane. Now what we are going to draw, I told you first only, the cell membrane is made up of what? Lipids. Just to draw in such a way. These lipids are arranged in bilayers. I draw the two layers like this. Just to simplify the diagram, not go complicated one. Okay, likewise is to draw the diagram. This is what we are going to call it as a, this is lipid, polar HED head. These are non-polar tail. This is about the non-polar tail, and this is what we used to call that as a lipid bilayer. Lipid bilayer. That is the lipid bilayer. Now what we are going to draw here, apart from the lipids, I told you the plasma membrane consists of what? Proteins and carbohydrates. Proteins and carbohydrates. Proteins and carbohydrates in the sense, I told you some proteins are classified into peripheral protein and integral protein. Now I used to draw like this. These are peripheral proteins, and sometimes you are going to get partially buried or buried within this, what we are going to call it as a integral proteins. What we used to call them as a peripheral protein as well as integral protein. What I told you, the proteins are of integral proteins. These are the integral proteins. Understood? And where we are going to get the cholesterol. Then glycolipids are also we are going to get in a plasma membrane. Glycolipids, glycoproteins, which are attached with the proteins. Some this is are now attached to the lipid, glycolipids. Then glycoproteins are also you are going to find in a what? Cell membrane. So I told you regarding the cell membrane. What else I told you? Cell membrane. The detailed structure of cell membrane was studied up to the discovery of electron microscope in 1972. And the cell membrane, structure of cell membrane was studied in a human organisms. The cell membrane consists of what I told you, lipids. These are the lipids, and they are arranged in a what? Bilayer. That is what I told you here, lipid bilayer. Outside the polar end and inside the non-polar tail. And these non-polar tail, those are going to protect from the aqueous environment. Then what I told you, the cell membrane is also consists of proteins and carbohydrates. The ratio of proteins and lipids is varies from what? One cell to another cell. And I told you in human organisms, the ratio of lipid, it is a 52% and proteins are other what 42. Sorry, in a human RBCs the plasma membrane consists of 52% of proteins and 40% of lipids. Then I told you the cell membrane consists of proteins and they may be what I told you two kinds of proteins. In the sense proteins are classified based upon their position in a cell membrane. If they are present on the membrane of a means surface of the cell membrane, we are going to call them as a peripheral protein, and if they are buried inside the cell membrane partially or totally, that time we are going to call that as an integral protein. That time we are going to call that one as a integral protein. Understood? That time we are going to call that as a integral protein. Then I told you about the improved model of this cell membrane that is discovered by Singer and Nicholson in 1972, and that improved model that is widely accepted is called as what? Fluid mosaic model. That is called as a fluid mosaic model. The question is going to ask you based upon this. What is the question? With neat label diagram, explain the fluid mosaic model of plasma membrane. Fluid mosaic model of plasma membrane, or write a note on plasma membrane. Then you are going to write whatever the points explained still for so far. Those points you are going to explain. Now, the few points I am going to write regarding the fluid mosaic model of plasma membrane, which is discovered by Singer and Nicholson. Singer and Nicholson. Okay. After the discovery of this electron microscope, I told you the basic structure, means basic component of a cell membrane, and that cell membrane is improved by the Singer and Nicholson, and they are given the name for it as a fluid mosaic model, and the fluid mosaic model of a cell membrane was widely accepted. What are the points? The few similar points are there, whatever we have discussed earlier. The fluid mosaic model of cell membrane consists of, again, the lipids bilayer, they are arranged in a bilayer. The proteins are embedded into the lipid bilayer of a phospholipids. Then polar heads, I told you, phospholipids face to the surface of lipid bilayer, and non-polar heads face towards the inner side. Then in between the lipid bilayer, number of proteins are present, and these proteins in a lipid bilayer, there are the three kinds of proteins you are going to find. I told you earlier what is peripheral and integral. Okay, in model or in improved Nicholson model of a fluid mosaic, you are going to get the transmembrane proteins outside the surface of the membrane, then intrinsic protein which are embedded into the two lipid bilayers, then extrinsic proteins which are exactly present on a what we are going to call them as a lipid membrane. In the sense like this, this is what I have drawn the diagram. First, and this is the diagram of lipid bilayer. Okay, and proteins which are present on a membrane, those are membrane surface, intrinsic protein, and the proteins which are present or embedded, those are, we are going to call that as proteins on a surface, transmembrane, proteins inside embedded, those are intrinsic proteins, and exact here on a what we are going to call that as a lipid membrane, these are what we are going to call as a extrinsic proteins. Those are called as a extrinsic proteins. These you are going to find in a fluid mosaic model or a improved model. And some glycoproteins and glycolipids bond to the membrane. In the sense, glycolipids and glycoprotein bonds are present on a membrane surface, act as a cell identity markers. That is, act as a cell identity markers. Those are act as a cell identity marker. This is what the explanation regarding the fluid mosaic model of Singer and Nicholson. It is to draw the diagram whatever I drawn earlier, and in that you are going to include these labelings. What are those? transmembrane, intrinsic proteins, extrinsic proteins, etc. Then you are going to write the glycoprotein and glycolipids. And as possible as you used to draw the simple diagram regarding the what cell membrane or a fluid mosaic model of cell membrane. And this is very important as a point of your final examination. In final examination, the question has to be asked based on this concept regarding the cell membrane, in the sense a fluid mosaic model of cell membrane. For 5 marks question with neat label diagram, explain the fluid mosaic model of cell membrane. That time you are going to draw the diagram. Afterwards, you are going to start with the fluid mosaic model was discovered by the Singer and Nicholson in 1972. The fluid mosaic model of cell membrane consists of, or it is composed of a lipids, and those are arranged in a lipid bilayer, and then you are going to write what we are going to call it as a these lipid bilayer is consist of the proteins are embedded into the lipid bilayers of a phospholipids. Polar heads of a phospholipids are faced towards the what? Lipid bilayer surface lipid bilayer, and non-polar heads are faced towards the inner side. Then in between the lipid bilayer, the number of proteins are present, and those proteins may be classified into transmembranes, intrinsic proteins, extrinsic proteins, etc. Then afterwards, in fluid mosaic model of cell membrane, you are going to find some glycoproteins and glycolipids bonds to the membrane surface. They are act as a cell identity markers. And you may include few points whatever I told you regarding the cell membrane first. Okay, before we used to start the fluid mosaic model. Now, after this, what else we are going to discuss regarding the cell membrane, in the sense last we used to discuss about the function of cell membrane. What is the function of cell membrane? Cell membrane consists of different types of proteins. Integral, in the sense transmembrane, intrinsic and extrinsic proteins. It consists of lipids. Okay, that is what phospholipids, and those are having the two ends. One is a head, another one is tail. And head, whatever I told you, the lipid bilayer, that is a polar and non-polar and all. So based upon the structure and its chemical composition, the plasma membrane plays a very important role in a passive transport. Passive transport, in the sense the transport of material from outside to the inside the cell and inside to the outside the cell based upon their concentration gradient. There any energy is not required. In the sense, the plasma membrane allow some particles or something, some ions passing through it inside the cell and outside the cell with the help of passive transport. And in some case, in passive transport, what is going to happen? The simple diffusion along with the concentration gradient, I told you, then facilitate the diffusion. That is the first function of what we are going to call it as a cell membrane. What is the function of cell membrane? First function of cell membrane is it is going to allow or facilitate the transfer of molecules. In the sense, many molecules can move briefly across the membrane. Passive transport. First one is passive transport. First one is passive transport. In the sense, many molecules are passed, or many molecules can move across the plasma membrane. Across the plasma membrane or a cell membrane. Across the plasma membrane, in the sense inside the cell and outside the cell. And whatever the passive transport is there, it is due to simple diffusion. Based on concentration gradient. In the sense what? Concentration gradient. concentration gradient, in the sense from lower concentration to higher concentration. Okay, simple diffusion. Another one is facilitated through diffusion. Okay, this is about the passive transport. In the sense plasma membrane allow to pass many molecules across the plasma membrane with the help of passive transport. Then some molecules can pass through the plasma membrane with the help of active transport. In the sense the active transport require energy in the form of ATP energy and all. Right? Here the molecules are what pass against the concentration gradient. The molecules has to pass through the plasma membrane against the what we are going to call it as a plasma membrane or cell membrane against the concentration gradient, and it requires the what active means energy, that is what active transport. Next one is plasma membrane osmosis. OSMOSIS. Here osmosis in the sense again the molecules, some movement of solvent or a water molecules from the area to the higher concentration to the lower concentration through the semi-permeable membrane. Higher concentration to the lower concentration through the semi-permeable membrane. In the sense this plasma membrane is act as a semi-permeable membrane. In the sense it is going to allow a particular molecules, and they are going to restrict some molecules passing through the plasma membrane. In the sense this is the main what we are going to call that one as a functions of a plasma membrane. Apart from this, plasma membrane act as a phagocytosis. Phagocytosis in the sense it is going to engulfing and digesting the solid food particles by the cell. Then next panacocytosis, cell drinking process, absorption of a fluid liquid droplets into the cell. Then exocytosis in the sense throwing out some what we are going to call it as a extracellular materials from the vesicles through the plasma membrane. So these are the what few functions regarding the plasma membrane. So you have to remember regarding the organelle, that is what plasma membrane. In plasma membrane I told you, cell membrane. Cell membrane I told you the different components which is made up of. Afterwards we will discuss regarding the Singer and Nicholson model. We call it as a fluid mosaic model. Points regarding the fluid mosaic model. Then we will discuss the functions of cell membrane. And the 5 marks question is going to ask you for the final exam, that is with neat label diagram explain the structure of fluid mosaic model. Explain the structure of fluid mosaic model. With this I am going to stop here, and I will continue in the next class with the next organelle, that is endoplasmic reticulum, or means with the next organelle. Okay, thank you.